Good day, listeners. Today we are going to learn about the criteria for algal classification, and I am Dr. Anami Augustus Arul, Assistant Professor, Department of Botany, Holy Cross College. Algae are diverse group of photosynthetic organisms that can be found in various environments like sea, that is in marine habitat, fresh waters, that is in ponds, streams, as well as in moist soil. Algae are simple plant-like organisms that can be unicellular or multicellular and come in a wide range of sizes and shapes. Algae play a crucial role in the ecosystem as primary producers, converting sunlight into energy through the process of photosynthesis. So these are the criteria for classification. Number 1, nuclear organization. Number 2, nature of cell wall components. Number 3, pigmentation and photosynthetic apparatus. Number 4, nature of reserve food. Number 5, flagellation. And number 6, type of life cycle and reproduction. First, we are going to see about nuclear organization. There are two types of nuclear organization. Either it can be prokaryote or eukaryote. So prokaryote means the cell which do not have any proper nucleus. So these nucleus are called as nucleoid because they don't have any nuclear membrane around the nucleus. So here in algae, the cyanophyceae or blue-green algae are the prokaryotic in nature, and all the other classes comes under eukaryotic condition. The next one is about nature of cell components. So the nature of the cell components, usually it will be polysaccharide. In some cases, it is lipid and protein. The inner layer usually will be cellulose, which is insoluble polysaccharide. Here you can see the structure of the polysaccharide. In algae, the chlorophyceae member will have cellulose as a cell wall, and xanthophyceae will have pectin substances. Chrysophyceae is having non-cellulosic substances which is silicified or calcified, and Phaeophyceae it is alginic acid and fucinic acid, and in Rhodophyceae it is non-cellulosic polysaccharides like xylans and galactans. The last one, that is Cyanophyceae or blue-green algae, the cell wall is more like bacteria and is made up of mucopeptides. The next one is pigmentation and photosynthetic apparatus. So you know there are lots of pigments like chlorophyll, carotenoids, xanthophylls, bilirubins, like that it's there. So here you can see various types of pigments, that is anthocyanins, hydroxy anthocyanins, melanin, oxopolyneins, quinones, carotenoids, like that. So in algae, usually the chlorophyll will be of either A, B, C, D and E, and in particular chlorophyll A will be present in all the algae. Chlorophyll B will be present in Chlorophyceae and Eugleniophyceae. Chlorophyll C is found in Phaeophyceae and Cryptophyceae. Chlorophyll D is found in Rhodophyceae. And chlorophyll E is confined to Tribonema of Xanthophyceae. And coming to carotenoids and xanthophylls, among the carotenoids, beta carotene is found in all the classes of algae. Alpha carotene is found in Rhodophyceae. Gamma carotene and lycopene are found in Chlorophyceae. E carotene is present in Basilarioophyceae. And there are about 20 types of xanthophylls commonly found in algae, like neoxanthin, neofucoxanthin, fucoxanthin, chetoxanthin, siphonoxanthin and oscillatoxanthin. Coming to the below proteins, they are water-soluble pigment and can be phycocyanin, phycoerythrin or allophycocyanin. In rhodophyceae, it is R-phycoerythrin, and in cyanophyceae, it is C-phycocyanin. And thylakoid also shows some variations. In chlorophyceae, there are 2 to 6 thylakoids per granum, and the pyrenoid are covered with starch plates. In xanthophyceae, phaeophyceae and basilarophyceae, there are 3 thylakoids per granum, and the pyrenoids are without starch plates. In rhodophyceae, thylakoids are single and widely separated in chromatophores, and pyrenoids are naked. And in cyanophyceae, the thylakoids are free in cytoplasm as chloroplast, are not found, and pyrenoids are also absent. The next is about the nature of reserve food material. The primary product of the photosynthesis is starch, so that is present in all the algae. And in chlorophyceae, it is exclusively starch, and in xanthophyceae, apart from starch, it will have the food product as oil and leucosine. And in rhodophyceae, it is rhodophycean or floridian starch, and in cyanophyceae, it is mixophycean starch, and in phaeophyceae, it is laminarian or mannitol. The next one is about the flagellation. So number and position of flagella are very important in segregating or differentiating the classes of algae. So in cyanophyceae and rhodophyceae, flagella are completely absent in vegetative as well as in the reproductive structures. In all other classes, the basic flagellar structure is similar. The flagella have 9+2 pattern of component fibrils. The flagella can be acronematic or whiplash, or pleromematic or tinsel and parsinate in type. In chlorophyceae, flagella are 2, 4 or indefinite in number. It is apical or subapical in position and acronematic type, that is isoconotic. In xanthophyceae, flagella are 2 unequal apical, 1 acronematic and 1 pantonematic. In phaeophyceae, flagella are 2 lateral, 1 acronematic and 1 pantonematic and unequal in size. In parsinophyceae, parsinate type of flagella are found. Next one is about type of life cycle and reproduction. Sexual reproduction, that is isogamy, anisogamy and oogamy, can be seen in the algae. Haplontic life cycle, diplontic life cycle and triphasic life cycle are characteristics of different groups. Sexual reproduction is completely absent in cyanophyceae. The reproduction is oogamous and life cycles are usually complex in rhodophyceae and phaeophyceae. In chlorophyceae, the reproduction can be isogamous, anisogamous and oogamous. The life cycle can be simple or complex. Altogether, you can see in this table about the diagnostic features of different classes. So basically, the divisions are chlorophyceae, xanthophyceae, basilarophyceae, phaeophyceae, rhodophyceae and cyanophyceae. And you can see the nuclear organization, the cell wall components, photosynthetic apparatus, storage products, flagellation and pigmentation. So by these features, the classes or the criteria for classification is analyzed. Thank you, learners. Have a good day.